Now I'm going to show you. So now we characterize very well the cellular process in the wild type. And we can also, and I show you that we can also study females. And if you see, this is the homologous region to the sex chromosome. Yeah, and you find a row of cells that don't rotate. You can also, we can also study mutants that have a diagonal uh, orientation. So if you see here, this will have more or less 45 degrees, and you can compare. Ask the question, what happened with the cell bed, what happened with the changes in apical cell area, and we can also study sex chromosomes of different lengths. So we already study the wild type, but we can also artificially select, and every year, every generation, for a year, what we did is select for combs that they were small, and in just 1 year, we made a comb that it was only 4 to 7 bristles, while the normal is 9 to 11. And we also were able to increase the number of bristles, sexcom teeth, by artificially selecting every generation. We count the number of bristles and select for the flies that have the longest sexcom, and we were able to get these flies that have 12 to 16 bristles, sexcom teeth, and try to study the cell dynamics to again try to test what are the processes that are unique of the rotation and what are other processes that depend on the degree of rotation or depend on the sexcom length. So I'm going to not show you all the results to not make this very long, but I'm going to show you the most significant results. Here is the movie here of the female. Here's a movie of a diagonal sexcom of a mutant, and here is the wild type. So as you see here, these row of cells don't rotate, and you don't see a lot of expansion. Here, this one rotate just a little bit, and they don't continue rotating at the end of the movie. And this one start, and the wild type, on the other hand, complete the rotation. And this movie shows combs of different sexcom lengths. So here we have the normal one. Here we have the small one with 5 cells, only 5 sexcom teeth. Here we have a sexcom that is very long, the long combs. And here we have a mutant that have long combs. And what we did is try to analyze what happened with the degree of cell expansion, what happened with how the cells mixed, what happened with the cell death. And I'm going to summarize all these answers in few slides. So in the following figure, I'm going to show you what happened with the changes in cell area, in apical cell area. And we're going to compare again these 4 lines, these small combs, the normal comb, the long combs, and this mutant that have a diagonal orientation in the comb. And we found that only the degree of apical expansion is similar between sex comb of different length. Here, the pink represent the distal region and the blue represent the proximal region, the region that is constricting, and this is the region that is expanding, the region that is growing, and the region that is getting smaller. Here are the changes in apical cell area. So when the changes in apical, the delta of the changes in apical cell area, and when the changes are the delta of the changes are positive, there's expansion, so all of them are growing. And when it's negative, there's a constriction. And if you see, all of them, as we previously said, all of them are constricting. But if you compare, the expansion and the constriction seems to be different. While if you compare the expansion, the degree of expansion, although there is some variation, all of them seems to be statistically homogeneous. On the other hand, when you have combs that are smaller or longer, the degree of constriction, the blue region, seems to differ, seems to be different. In particular, the small and the long one seems to be significantly different. So this process seems to be the degree of constriction seems to vary a lot and possible, and is again heterogeneous, while this one is the most homogeneous process and is a good candidate to be responsible for the rotation. So to summarize, as I said before, we have multiple processes happening. Instead of having one cellular process, we find many more cellular processes, and I show you the data between 23 to 36 hours after pupation. Before, there are other processes taking place, including cell proliferation, but the comb doesn't rotate at that moment. So we mainly focus on this one. It seems to be the most relevant for the rotation. And I'm going to show you, I'm not going to talk in detail about every single cellular process, but I'm going to show you a schematic to show you, for example, what happened with the cell extrusion, to show you that is very heterogeneous. So for example, when we compare the combs that are small, the combs that are normal in size, and the combs that are long, we found that the cell extrusion takes place above the proximal region, about this region, while in the wild type, most of the extrusion in the movies that we analyze is located in this region, while in the combs that are very long, some of them show the extrusion in above the comb, while other ones they are located very close to the comb. So in other words, there is every time that we study the cellular process, the other cellular process, the cell extrusion, the interchanging of cell neighbors, the interchange of cell neighbors underneath the comb, all this process seems to be very variable. Even the reduction in apical cell area, all of this one seems to be very, very variable. The only process that seems to occur only during the rotation seems to be the increase in apical cell area. For that reason, our current hypothesis is that the data that we have is best explained by the push model. So again, and as I started in the beginning, we found that the cells are increasing here in apical cell area, while these ones are reducing in apical cell area. And this allows us to propose three models that we discussed in detail previously. And from those models, the one that seems to be that best fit the data is the push model, because as I show you, this in the blue section, the proximal above the comb, the cellular processes to be highly heterogeneous. There is a lot of variation, while the changes in apical cell area seems to be very homogeneous. And this seems to be consistent with the push model, that the changes in apical cell area are producing a push, while what is happening above the comb is just a passive response of the rotation.